Hello, my name is Natalie Verkade and this is my presentation on Grehlin. In its active form, Grehlin has been identified as a 28 amino acid peptide. Most of it is secreted by specialized cells in the stomach, enteroendocrine cells, and these are scattered in the gastrointestinal tract mucosa. Most of the remaining secretion originates in the small intestine, although there are some tissues outside the GI tract that have been shown to secrete Grehlin. Grehlin is known as the hunger hormone as it's secreted in response to fasting and acts as a strong appetite stimulant. Levels increase prior to eating and decrease once you are in a fed state in a normal, healthy individual. The initial stimulus for Grehlin secretion is an unstretched, empty stomach. Grehlin is released in the stomach and this stimulates hunger by influencing sensors in the hypothalamus that regulate appetite control and feeding behavior by stimulating the secretion of the neurotransmitter neuropeptide Y. Locally in the stomach, Grehlin acts as a paracrine hormone, modulating the GI hormones that control gastric emptying and motility. The maintenance of energy homeostasis is dependent on the balance between energy intake and expenditure. Once mechanoreceptors in the stomach are stretched, secretion of Grehlin ceases, and secretion of leptin by adipose tissue indicating satiety commences. Interestingly, the receptors for the opposing forces of Grehlin and leptin promoting energy homeostasis are located on the same cells in the hypothalamus. Once Grehlin is synthesized, mostly in the stomach as a peptide hormone, it is transported in the blood dissolved in plasma. On reaching the target cells, it binds to a G protein coupled receptor. This directly activates the amplifier enzyme phospholipase C. This in turn activates second messenger systems of IP3 to release intracellular stores of calcium and DAG to increase phosphorylation of AMP-activated protein kinase. Together, the phosphorylation and the increase of calcium produces the cellular response. Grehlin is not only responsible for increasing appetite, it stimulates the release of growth hormone by the anterior pituitary. There are both starvation and growth pathways of Grehlin. As we know, Grehlin stimulates food uptake, helping to maintain homeostasis. By acting on the hypothalamus and directly on the anterior pituitary, it also increases secretion of growth hormone, which promotes protein synthesis to allow tissue growth, mobilizes fats from adipose tissue, and decreases glucose uptake by inhibiting insulin and therefore conserving glucose. So as we can see, Grehlin interacts with other systems, including the pancreatic hormones, most notably insulin. As we know, Grehlin levels increase during fasting in the plasma, and this means that it inhibits the uptake of glucose by the cells, promoting glucose sparing. In obese individuals, there is a reduction in circulating Grehlin levels, and this may explain why they have a higher excess of circulating insulin in relation to circulating glucose in their system. So as we can see, Grehlin interacts with all other systems, as do most of the hormonal systems, endocrine systems in the body. Prader-Willi syndrome is perhaps one of the best known syndromes connected with excessive Grehlin secretion. It's a rare genetic order that it appears from birth. It begins off with infantile hypotonia and developmental delay and can lead to mental retardation and behavioral disorders. It tends to lead to a high level of obesity and presents itself between 1 to 6 years of age. Sufferers of Prader-Willi syndrome have very abnormal food-seeking behavior. They hoard food and they also eat spoiled or rotten food. They steal food. They're known to have to put locks on pantries and refrigerators. It is thought that excessive secretion of Grehlin plays a role in this syndrome, as there is no off switch in that the levels of Grehlin do not decrease after feeding. Therefore, syndrome sufferers are always hungry and are always seeking food. Further studies have shown that appetite stimulating properties of Grehlin are separate and independent from the secretion of growth hormone, as Prader-Willi syndrome sufferers often have short stature. However, the excessive secretion of ghrelin and the fact that it doesn't decrease after meals leads to the overeating, and this is consistent with the idea that ghrelin is a physiological regulator of appetite in humans, and it is independent of its secretory role, stimulating role in the secretion of growth hormone. In Prader-Willi syndrome, the physiological effects are quite apparent. Excessive ghrelin secretion leads to an appetite which is disproportionate to normal levels. Ghrelin levels do not fall after a meal, so Prader-Willi syndrome sufferers are always hungry and seeking food. Current treatment is the human growth hormone, and this further supports ghrelin's role in stimulating appetite is independent of its stimulation of growth hormone secretion. The role of ghrelin in Prader-Willi syndrome has not yet been firmly established, although it is apparent that plasma levels of ghrelin are generally elevated in Prader-Willi syndrome sufferers.